Thank you. We're going to take questions at the end of the session for the group. So we'll pass straight on to the next speaker, who's Jim Stevens from Eli Lilly. And I'd remind you all again, the bios are all in the packet that you got when you checked in. Jim.
Well, thank you very much. Pleasure to be here. I was at a Gordon conference this summer, and one of the speakers was talking about science and public policy. And she was going on and on about a Pew Research Foundation poll looking at what are the most trusted professions and how scientists were among the most trusted professions. She said it over and over, and then she stopped briefly and said, oh, by the way, except for you guys in industry. So as the least trustworthy person on the program this morning, I'm still going to try to make some provocative statements, I hope. What is the problem statement? I'm going to talk about drug safety. Eventually, when you're talking about regulatory science, you come around to drug safety because it's such an important problem. I'm not going to read this statement for you, but it's taken from the Reagan-Udall Foundation website and the Predict Tox problem statement of why we need to do this. And what I'm going to do is just take some of these highlighted blue phrases, and I'm putting the same phrases on this slide, and I've now color-coded them. Why do that? The reason for doing that is they exist in different domains. Everything in blue here is a technology. Everything in red here is a task, improving patient safety, increasing efficiency. Everything in green is an organization. And the people part is the folks in this room. Why do that? This is Harold Levitt. Harold Levitt was a psychologist who was very interested in how organizations function and organizational change. And what he proposed is that these domains of structure, task, people, and technology are highly interdependent. And not only are they interdependent, if you don't get them all right, you will not make progress. And if you get them wrong, you can actually cause negative tension within the organization. So if you ask people to do things they can't do because they lack technologies, you get negative tension in the organization. If the organization doesn't propose the right tasks, then people can use technology, but they can't actually make any progress. So what do these organizations and these tasks mean for how we use new information and new technology to make progress in regulatory science? So I'm going to talk about two things. I'm going to talk about what we do today, and I've kind of faded out what we're going to do in the future. I'll come back to that in just a minute. Okay, complicated slide. Let's look at how we do safety assessment today, and let me break it down for you, as Martin said, unpack it just a little bit. It's a series of steps. At the top, we have a test, a non-clinical test. In the middle, we have a clinical test, and then we have two steps where we make a judgment. What I'm going to try to emphasize is we have tests and we have judgments. I've defined non-clinical toxicity as NT, and you either get it or you don't get it. And at the bottom, we're trying to say, is it safe or is it not safe? That's a judgment. This is an assay result. If we don't get non-clinical toxicity, we then go to the clinic. And we ask, was it toxic clinically or not toxic clinically? If it's not toxic clinically, we continue and you say, it's a true negative. It's safe. I'm leaving efficacy off the table for the moment. If we see something that we don't like, we get a clinical toxicity, now we have to say, is that acceptable? Is it nausea and vomiting, or is it irreversible cardiac injury? If it's acceptable within the disease context, we say, well, yes, we can actually continue. If it's unacceptable, we terminate. Now, this is a technical definition. This is a false negative. This is a true negative. We have both false negatives and true negatives, which are safe, and we have false negatives which are not safe. This is more like what we do on a regular basis. We always push these non-clinical tests to produce the toxicities, kind of like an engineer. You push the system hard enough to break it and see how it breaks, and then you try to improve on what breaks if you put the system under an extraordinary load. What we do is we say, is it monitorable, reversible? What's the margin of safety? What's the disease context? And we make a decision. Is it manageable? Is it okay to put people at risk knowing what we know? And if we say it's not, we say it's not safe. Now notice, we never know the outcome. All we have is the test result. We never know the outcome in the clinic. But if we say, yes, it's manageable, we go into the clinic and we say, well, we didn't see what we expected, so it's a false positive. We did see what we expected, it's a true positive. Now we have to make, and if it's a false positive, we continue. If it's a true positive, we got to make one of those acceptability decisions again. Is it okay to get bone marrow toxicity in the context of an oncology clinical trial? The answer is almost always yes. But there may be outcomes that we underestimated the risk, and it is not acceptable, and we terminate. Now, let's add these up. I'm going to make a very simple point. We have safe, which are true positives and false positives, not safe, which are true positives and false negatives, and we have safe, which are true negatives and false negatives. If I'm establishing a confusion matrix approach to estimating the performance of this test, I'm going to focus right here. But this is what we see. You can't confuse understanding the performance of a system from a technical basis with the performance of a system from a judgment-based perspective. We do that all the time when we think about regulatory science and safety. So what are the points? Avoid data-driven errors with new technologies, but avoid judgment-driven errors by understanding the tasks. What kind of information do you need to do both? Remove organizational barriers to use better information, provide training and incentive to use information to adopt new approaches to tasks. Now, I'm going to submit to you, and we've thought about this a lot, no one actually knows the current technical performance of non-clinical safety testing, yet everyone wants to improve it. And I will submit to you, no one in this room can actually provide me an adequate measure of how much we improved or if we're doing as well as we used to do if we changed the system. Okay, let's move on and talk about the future. 2 more tasks: predict adverse events, understanding multi-scale complexity, systems pharmacology, computational modeling, and I put in parentheses networks, which is also from the Reagan-Udall website. I've been doing gene expression since you used to collect gene expression data with little spots on audio-radiographic film. When I send some of my papers around, the students really laugh at me. It's amazing to be doing gene expression work and feel like a dinosaur. This is a multi-scale problem. This is a proteasome. This is a human being. We're looking at scales of complexity that are mammoth when we try to extrapolate from what happens in a biochemical assay to what happens in a human being. This is a gene expression experiment. Dimensions of time and dose. The only thing you probably recognize about this gene expression experiment is that green means down and red means up. These are 415 unsupervised co-regulated networks that have been extracted from 20,000 individual measures over 1,000 different experiments. We've reduced the dimensionality of this data set almost 99% and retained all the biological information. Now, if I ask everyone in the room to say, at which dose, low, medium, and high, did something happen? And at which time was it maximal? I think everybody in the room could make that conclusion. And if I ask you, in which one was there a short-term effect that actually went away, and at which one was there a short-term effect that didn't go away, you can make that conclusion. You have pathology here, you do not have pathology there. What you see on the right are the effect sizes for the association of change of any individual network with the occurrence of pathology either at a future time or co-occurring. So I can read this and say, this is highly associated with pathology, these are all up, it's likely I had pathology. And by the way, the proteasome, which was inhibited by bortezomib in this experiment, in the rat liver, is right there. So now I'm looking at induction of the proteasome component genes simply by inhibiting the proteasome in the context of everything else that happens in the cell. I love this depiction. What's good about it is it puts high content data, uses data reduction methodologies, and puts it in a pattern recognition format that I can use as a toxicologist to make a safety decision. You can't do this from an Excel spreadsheet. You can't do this from a heat map. You've got to take high content data and put it in ways where people can actually make decisions. By the way, this is a map for hypergeometric enrichment of known pathways. Red means there's a lot of information there. And by the way, that's the proteasome right there, if you can see my spot. Everything down here is most associated with the occurrence of toxicity. Look at the big blank spot. What that says is the things we know in pathway databases are the things that are least likely to be associated in an unsupervised way with gene expression networks that are associated with pathology. There's stuff we don't know. All right. How do we improve? We need scalability across dimensions. We need to interpret data in a context of prior and future experience. We need to be able to link known and unknown biology with safety outcome. I'm going to apologize for sounding like Donald Rumsfeld here, but knowing what we know and knowing what we don't know is really, really important. And the data that tells us that is the most important information. And we need to look at safety as an evolutionary biology problem. If I look at networks and how they're conserved across evolution, I can reduce safety to a function. I can say the likelihood that something that occurs in an animal test will occur in a human is based on three things. How much did it change? What was the association with pathology? And how well is that network conserved across species? But we don't do that on a regular basis with gene expression data. All right, summing up. Don't confuse judgment-based errors with data-driven errors. Focus on information that's necessary for the task. Bigger is not always better. Organizational change management is critical to achieve transformation. The most interesting thing I do is try to do better safety predictions with gene expression. The most frustrating thing I do is try to get people to use it. Systems-level approaches must be scalable, integrate data into biological context, and reduce dimensionality. I have no idea if I was on time, but if I was, thank you very much, and we'll take questions during the panel.